everyone, in this lesson I'm going to talk about electron transport and oxidative phosphorylation. So in my previous lesson I talked about electron transport chain, and we learned that when NADH and FADH2 are oxidized, they loses their electrons, and these liberated electrons are then traveling through the electron transport chain. That means they travel from complex 1, complex 3 all the way down to the final electron acceptor, that is oxygen. And during the process, the protons from the mitochondrial matrix are pumped out into the intermembrane space, and eventually it will create these proton gradients. Basically what it means that more protons are being driven out from the mitochondrial matrix into the intermembrane space. And this also creates an electrical gradient with more positive charges on the outside of the membrane, which is the intermembrane space, whereas the matrix becomes more negative. And furthermore, it also creates a pH gradient where the outside of the membrane has low pH, whereas the mitochondrial matrix has high pH. So once the proton gradient is established, the proton from the intermembrane space then diffuses down its electrochemical gradient through complex 5. As they are diffusing through complex 5, ADP is phosphorylated to synthesize ATP. So basically the important message here is the formation of proton gradient. If the proton gradient is not established, that means the protons cannot diffuse through complex 5, and that can lead to the inhibition of ATP synthesis. So the question is, how does a proton gradient drive the synthesis of ATP? And before we learn how the ATP is synthesized, we first need to learn about what is complex 5 and what is the structure of this enzyme. So complex 5 is basically also known as ATP synthase enzyme. And ATP synthase is a large complex enzyme that looks like a ball on a stick located in the inner mitochondrial membrane. And much of the stick part, which is also called the F0 complex, is embedded in the inner mitochondrial membrane, whereas the ball part, which is called the F1 complex, which protrudes into the mitochondrial matrix. So F1 complex consists of A, B, and C subunits. So C subunit, which is also called as C ring, which is here in the orange color, is embedded in the membrane, and which also comprises this like the cylindrical structure. So they have this kind of 18 to 15 subunits. And A subunit, which is in the yellow color, which binds to the outside of the C ring or the C subunit. And it also has this B subunit, which is in the green color, which has an extended arm which is connecting to the F1 complex. Now, F1 complex consists of 3 alpha, 3 beta, 1 gamma, 1 sigma, and 1 epsilon subunits. 3 alpha and 3 beta subunits, they are arranged alternatively and they form this hexameric ring. And it is also the catalytic subunit of the ATP synthase enzyme, meaning it catalyzing the reaction to synthesize ATP. Now F1 complex also has this one gamma subunit, which is in the gray color. It literally looks like a stick. One side of the gamma subunit is connected to the C subunit of the F0 complex, and on the other side it is connected to the hexameric ring of alpha and beta subunit of the F1 complex, meaning that it actually is important in connecting F0 and F1 complex. Now it also has this one delta subunit, which is in the brown color here. It actually caps the hexameric ring of the alpha and beta subunits. And finally, it also has this one epsilon subunit, which associates with gamma subunit and C subunits. Now let's talk about how is ATP synthesized through complex 5 or ATP synthase enzyme. So once the proton gradient is established, protons from the intermembrane space enter the inlet half channel. So once inside, protons then pass through the C subunit until they reach the outlet half channel, and once they reach the outlet half channel, protons then exit into the mitochondrial matrix. And when the protons are passing through the C subunit, it actually causes the rotation of this C subunit. Now the gamma subunit, which is in gray color here, which is connected to the C subunit, so the rotation of the C subunit also causes the rotation of the gamma subunit. Now gamma subunit is also connected to the center of the hexameric ring, which consists of alpha and beta subunit. However, the hexameric ring of alpha and beta subunits is stationary, it cannot move. So when the gamma subunit rotates, it actually comes in contact with different beta subunits, and that causes the conformational change in beta subunits, and this conformational change in beta subunit results in the phosphorylation of ADP to make ATP. So that means conformational change in beta subunit is crucial here to synthesize ATP. And as I mentioned earlier, that hexameric ring forms the catalytic subunit of the ATP synthase enzyme. However, if to be more specific, it is actually the beta subunit which acts as a catalytic subunit than alpha subunit, because changes in conformational change in beta subunit causes the ADP phosphorylation to synthesize ATP and not the alpha subunit. So this is actually a very simplified version of ATP synthesis through complex 5. It is actually more complex, and I will definitely be making a video on mechanism of ATP synthesis through complex 5. So to summarize the steps of ATP synthesis, we learn that protons flow through the F0 complex or F0 cylinder, and when they flow, it causes the C unit or the C ring to rotate, and movement of this C ring causes the gamma stick to rotate, and gamma actually comes in contact with the different beta subunits, and that causes the conformational change in the beta subunit, and that results in the phosphorylation of ADP to make ATP. And one important thing to remember, that it requires 4 protons to synthesize one ATP molecule. And this way of synthesizing ATP through complex 5 is called rotational catalysis. Rotational because it is when the protons are diffusing through the complex 5, it causes the rotation of C subunits and gamma subunits, and then it comes in contact with the different beta subunit, which then catalyzes the reaction to synthesize ATP. Therefore, it is called rotational catalysis. Now, I also want to talk about the number of ATP generated from one NADH as the electron donor. So we learned that when NADH is oxidized, it then loses electrons to electron transport chain, and these electrons are then traveling from complex 1, complex 2 all the way down to oxygen, and during the process, the protons are then pumped out from mitochondrial matrix into the intermembrane space. The number of ATP synthesized is calculated based on the PO ratio, and the P has to do with the number of phosphorylation of ADP to make ATP from one oxygen molecule consumed. So when one NADH is oxidized, basically there are 10 protons which are pumped out into the intermembrane space, and out of 10, 4 protons must be used to make one ATP. So if you take the ratio, out of 10 protons are pumped out and 4 protons are required to make 1 ATP, so that would give you 2.5 ATP. So if you get rid of the decimal, it will give you 3 ATP. So that means 1 NADH gives you 2.5 or 3 ATP. Now let's talk about FADH2. So when FADH2 is oxidized, the electrons are traveling from electron transport chain all the way down to oxygen molecule, which is the final electron acceptor. So when NADH is oxidized, there are basically 6 protons which are pumped out into the intermembrane space. So if you take the ratio, 6 protons are pumped out and 4 protons can be used to make 1 ATP, so that will give you 1.5 or 2 ATP. So that means 1 FADH2 gives you 1.5 or 2 ATP. Now let's also tally how many ATP synthesized from one complete oxidation of glucose molecule. So first is the glycolysis, and if you remember that glycolysis takes place in the cytosol, and which gives you 2 NADH. And we just learned that 1 NADH gives 2.5 ATP. So 2.5 multiplied by 2 gives you 5 ATP. However, I have written 3/5 because remember that this NADH is generated in the cytosol and electron transport chain takes place in the mitochondrial matrix. That means this NADH must be transferred into the mitochondrial matrix for it to be oxidized. And there are 2 pathways which play an important role in transporting NADH from the cytosol into mitochondrial matrix. One is malate aspartate shuttle and second is glycerol 3-phosphate shuttle. So I will definitely be having a video on those pathways where you will learn that how NADH from the cytosol is transported into the mitochondrial matrix. And depending on what pathway has been used to transport NADH from the cytosol into mitochondrial matrix, it can either generate 3 or 5 ATP. That's why I have written 3/5. And glycolysis also generates 2 molecules of ATP. So here you have 2 ATP molecules. And second is the pyruvate oxidation, which also generates 2 NADH, and that gives you total 5 ATP. Lastly, this is the acetyl coenzyme A oxidation, that is also called a citric acid cycle, and it also takes place in the mitochondrial matrix. So citric acid cycle generates 6 NADH, which gives you 15 ATP, 2 NADH, 2 FADH2, which gives you 3 ATP, and 2 ATP through substrate level phosphorylation. Total all this, you would get 30 or 32 ATP from complete oxidation of one glucose molecule. Now, in other places, you would find that one NADH generates 3 ATP or one FADH2 generates 2 ATP. So if you get rid of the decimal, it will give you 3 ATP or 2 ATP. And if you tally accordingly where you would consider 3 ATP per 1 NADH and 2 ATP per FADH2, it will give you either 36 or 38 ATP. And you will find this in many places. So I thought it would be just nice to give you a comparison that it is not wrong. You will find this in many different places, and do not get confused that why somewhere it has mentioned 30 or 32, other places it has mentioned 36 or 38 ATP. It is all right. So far we have learned that this is the electron transport chain, and where FADH2 and NADH are oxidized, and the electrons are then travel through electron transport chain all the way down to the final electron acceptor, that is oxygen. And in the process, protons are being pumped out into the intermembrane space, creating this proton gradient. And once the proton gradient is established, these protons are then diffusing through complex 5 to synthesize ATP. And this way of synthesizing ATP, where protons are diffusing down its electrochemical gradient, is called chemiosmosis. And when you combine this electron transport chain and chemiosmosis, it gives you oxidative phosphorylation. So first part is oxidation, where NADH and FADH2 are oxidized and loses their electrons. And the second part, where ADP is phosphorylated to synthesize ATP. So combined, they are called oxidative phosphorylation. And one important part here is the formation of this proton gradient. If the proton gradient is not established, it can then lead to the inhibition of ATP synthase and ATP synthesis. So basically, proton gradient couples the electron transfer to ADP phosphorylation to make ATP. Now, there are certain conditions where the proton gradient can be disrupted, and that can lead to the uncoupling of electron transport to ADP phosphorylation to make ATP. So basically, disruption of proton gradient results in the uncoupling, and during uncoupling, protons can leak through inner mitochondrial membrane to re-enter into mitochondrial matrix without channeling through complex 5 enzyme, and that can result in the inhibition of ATP synthesis. Now, there are some chemicals or compounds or proteins which can cause disruption in the proton gradient, and they are called uncouplers. And these uncouplers are categorized in different category. First is membrane damaging agent. To give you an example, they are RAC and AZT. So this uncoupler first is cytosine arabinoside, which is used in the treatment of cancer, and AZT, which is azidothymidine, which is used in the treatment of HIV. So these uncouplers actually break down the structural integrity of the mitochondrial membrane and renders the inner mitochondrial membrane permeable to protons. That means by damaging the membrane can allow protons to re-enter into mitochondrial matrix. Now, second category of uncoupler is mobile proton carrier. For example, we have DNP, which is dinitrophenol. So DNP actually binds to protons and transport proton across the mitochondrial membrane. Now, third is a proton channel. One such channel is UCP, uncoupling protein 1. And basically, this allows protons to pass through the inner mitochondrial membrane to re-enter into mitochondrial matrix. So basically, the main function of these uncouplers is to allow the protons to re-enter into mitochondrial matrix without energy being captured as ATP. In fact, the energy from this proton leak is released as heat, and it is also called as non-shivering thermogenesis. This is also the reason when RAC and AZT given in high dose, the patients actually suffer from fever because of the proton leak mechanism. It actually increases thermogenesis in their body. So there are three things to remember about uncoupling. First is acceleration of electron transport chain. So when the rate of electron transport is higher, more protons are being pumped out into the intermembrane space. However, as soon as the protons are in the intermembrane space, they then leak through the inner mitochondrial matrix instead of protons diffusing through complex 5, and that can result in the second point, which is the inhibition of ATP synthase enzyme and therefore inhibition of ATP synthesis. And third, because of this proton leak, it can also causes increase in heat production. Now I also quickly want to talk about uncoupling proteins, most importantly UCP1, which is also called as thermogenin. It produces heat in brown adipose tissue in mammals, and brown fat, unlike white fat, uses 90% of its respiratory energy for thermogenesis, in most importantly in neonates and in hibernating animals. This is also the reason that, for example, polar bear, they live in extreme cold weather, and during hibernation they need to maintain their body temperature, and these animals have high volume of brown fat, and that actually helps to maintain body temperature during this extreme cold weather. And the important fact is adult humans, unlike newborns, do not have much of brown fat. They have more white fat compared to brown fat. And now lastly, I quickly want to talk about some of the inhibitors of oxidative phosphorylation, and these are the inhibitors. They are used widely in research to examine the mitochondrial respiration. So the first one is the group of compound called amityl and rotinon. They actually inhibit the passage of electrons from complex 1 to coenzyme Q. Rotational is basically, it obtains from the roots of several species of plants, and aborigines in certain part of the world have made a practice of beating these roots of tree along the river bank to release this rotational into the water. And when rotational is released into the water, it then paralyzes all the fish and makes them easy prey for these people. Amityl, also known as amobarbital, also inhibits complex 1 enzyme of electron transport chain. Now second is antimycin A, which inhibit the passage of electrons from complex 3 to cytochrome C. And the another group is cyanide, sodium azide, and carbon monoxide, which inhibits complex 4. And last one is oligomycin, which basically binds to the F0 complex of complex 5. Basically, it inhibits the entry of protons to re-enter into mitochondrial matrix, and therefore therefore inhibiting the synthesis of ATP. So I thought this would be something interesting to share, because this is widely used in research, and sometimes if you are reading any research paper, you will come across some of this inhibitor used in the lab, and so that you feel familiar about these inhibitors. So if you learn anything new from this video, and I hope you really do, please like and share the video and subscribe the channel, and I will see you soon next time.